Welcome, dear students. Today we're going to start with studying the chapter number 5, and we're going to start with the DNA, and we need to get some information about it and a kind of the introduction of the episode, of the introduction of the chapter. Okay. DNA and genetic. Today we're going to start with the chapter number 5, and we need to discuss some information about the DNA and genetics. We need to study some information and some questions in our daily life about the DNA and how our bodies are designed by the Creator. Well, let's start. In the following lesson, we're going to study and discuss some fundamental or structural information and questions in our life, such as what? We need to know that what directs the single fertilized egg from which each of us originated. I mean, how we are, or from where we came from, and the resulting of mass of cell that grow up and up, that takes the shape and unique or rare individuals. Okay, and also we have some questions such as what makes each of us has a distinct or specific or definite shape and characters and personality from each other. And also we need also to discuss the similarities and differences between individuals or the different humans. All of these answers, today we're going to answer all these questions from our lesson today, which is genetic information. And also the units of genetic information that govern or that controls the inherited characters, which are called genes. Starting to learn more about our lesson. Starting with the biologists or the scientists of biology, they found that at any cell or each cell, it starts to divide, and after that, the chromosomes inside the nucleus. Try to remind you that any cell consists of two main parts, which is nucleus, and the nucleus is the manager, as we call it, or the main part inside the cell, and this nucleus surrounded by a cytoplasm, or a liquid which contains all organelles of each one of them has its own functions. Here the scientists found that chromosomes, they are found inside the nucleus, okay, and it can separate from each other and also contains a number of chromosomes. They are found in the original cells. And scientists considered that these chromosomes were a barrier of genetic information. The scientists, at the first they got confused, they got disturbance, which part inside the cell that carries the genetic information, which part inside the living organism cell that carry the information that reflects or represents our character, personality, everything physiological or psychological characters. Scientists at the first, they thought that the genetic information are carried by chromosomes, but chromosomes are composed of two main substances, DNA and proteins. In our lesson, we're going to start to study the structure of DNA, and in the next lesson, we're going to start how the DNA is synthesized and how the DNA is partitionized inside your body. Well, which of them carries the genetic information, DNA or protein? this is a problem and this is a question in the whole lesson that we're going to start. Which part in our body, DNA or proteins? Well, obviously, genes must contain a variety of information. For example, the scientists at the first, they thought that we have a little number of genes, but after that they discovered that we have a large and wide number of genes, and each gene, it has a clue or key that controls in a character inside our body. And if this gene has a problem or disorder, that may reflects by disease or by disorder and functionless of this organ. It was known that before that proteins are a diverse and complex group of molecules. They thought that before that proteins, they are a very complex or very large molecules. But what happened after that? They found also that proteins contain about 20 different kinds or types of amino acids. Amino acids, they are the structural and fundamental units that formed the whole structure of proteins. Again, we have each macromolecules, we have the monomolecules or a very tiny structural units, they are called monomers. Okay, so the monomers of proteins, which is amino acids, we have how many types of the amino acid that we have? We have 20 different types or kinds of amino acids. They are already responsible for forming the different in variety and all variation between the proteins. They actually different in their combination or the complexation. Where the DNA itself contains what? It contains 4 kinds of nucleotide or monomers. So we have here the question that we started by the episode. We said that the scientists, they got confused or disturbed between that the DNA is responsible for carrying genetic information or the proteins which carry the genetic information. I'm going to answer this question in our episode. And after that, we're going to study the structure of DNA and of protein to get more information about which part of them that already carries the genetic information. Within our speech, we said DNA, it consists of, or it includes 4 different types of monomers, each one of them called nucleotide, as we're going to know their names. While we said the proteins, it includes 20 different types or kinds of amino acids. Well, therefore, the scientists, they thought that at the first that proteins carried the genetic information depending on the quantity, not the quality of the genetic information. The scientists at the first, they thought that the number of amino acids is 20, so it necessarily carries our genetic information. Okay, but on the other hand, some scientists, they proved that the DNA it's already carries the genetic information of the human body. But in 1930, this was shown to be a wrong, that DNA contains the genetic information. The discovery or this invention that DNA is the genetic material, it leads for something that scientists, they need to study the molecular basis of inheritance, and also it called molecular biology. So after time, after 1930, by the increasing of technology and uprising much more devices to study the monomers and very tiny particles under microscopes, there is something which came out of blue, or the molecular biology. The molecular biology, actually, it's one of the newest science later that appears literally for studying the DNA and carrying the genetic information of our body. And also one of the most exciting and fast-growing fields of modern science. Well, and we have some evidences that DNA is a genetic material. What does it mean, evidence? Evidence, it's mean we have a clear-cut answer, that something that proves for us that already DNA is the genetic material that carries our genetic information, not the proteins. Let's start some of these experiments. Bacterial transformation. Well, bacterial transformation, actually, this is the first evidence or the first proof that the DNA is the genetic material or the material that carry the genetic material inside our body. Well, so this is the first evidence to cast a doubt on the belief that genes were made up of proteins. In 1928, when the British scientist Griffith, he studied a bacterial pneumonia. It's a species of bacteria, or it's a different specific type of bacteria. When he studied, or after his studying this type of bacterial, he discovered that the strain of pneumonia bacteria, okay, strain here it's mean race or a type or kind of bacteria. So we have a lot of different types of bacteria, okay. He discovered that this type of bacteria could be transformed into a genetically different strain. So at this time he has ability to transform or to change it genetically to a different strain, to another type, another kind of bacteria, but from the same species. And also the two strains he studied, okay, one of them called virulent or S-type, that killed the mice when he made his experiments on the mice, in which he had been injected. He brought a group of mice, and each one of them has been injected by this virulent type of bacteria, or the S type of bacteria. The other type, and we have here two types, okay, the virulent, and after time we're going to study the non-virulent bacteria, or the S type and R type. And we're going to know also the effect of S type or the virulent bacteria and non-virulent bacteria, okay. One of them is contained virus and the other one doesn't contain virus. And what gonna be happen if we infect the both type of the S and R types of bacteria to the mice? This is the experiment of the Griffith, we call it Griffith's experiment. It's a very famous in the genetic field. The other strain, which doesn't contain any viruses or no virulent and didn't kill the mice, or we call it R type. So we have a Griffith experiment. He brought two different types of or two different strains of bacteria pneumonia. One of them is virulent, and we call it what? S type or S kind. The other type, which is R, and he brought two groups of mice. The R have been injected by the S type, and the other group is injected by R type. And after that, the R type didn't kill the mice because there's no any infection. On the other hand, S type, the old mice, they are already had been killed. But if the virulent bacteria, which have been killed by heating, were injected into mice, what gonna be happen again? He supposed that we need to do some experiment, kind of documentation, and to get much more evidence about that the the DNA is already the main carrier for genetic information. Well, he supposed something that if the virulent bacteria, okay, which have been killed by heat, he exposed the bacteria to the heat, and after that he injected the mice by these killed or already destroyed bacteria. The mice, what happened? Didn't die. So when another group of mice were injected with non-living virulent bacteria, which is the killer one, S type, along with the living, parallel or the same time with the living non-virulent bacteria, some mice are died and the others didn't. So by examining the mice, he found that they contain living virulent bacteria or the S type. Again, he brought two groups of mice, and they have been injected by R and S types, and after that one of these types are subjected to the heat, and he repeats the experiment again, and by examining the mice he found that they contain a living virulent bacteria S, but they didn't die. Well, Griffith concluded, or he deduced, that some of the genetic material from the virulent bacteria had entered, okay, but the other type, the non-virulent ones, transformed them from non-virulent to virulent form. So this is a kind of transformation, or this type of changing and changing its character. This is experiment of the Griffith, or the first experiment about the DNA or the genetic information. Well, this phenomenon was named by bacterial transformation, or that the bacteria has ability to transform or to change its strain or its type from virulent to non-virulent and vice versa. And also he didn't explain, or he didn't give a final answer of how the genetic material transformed from S-type bacterial to R-type, or vice versa, from virulent to non-virulent and so on. So he gave us a final cut answer about the bacterial transformation, or the genetic information, genetic material. It has ability to transform, and he gave us an example, which is virulent and non-virulent bacteria. But he didn't give us the explanation or justification of what happened for the genetic material to transform from S-type to R-type, or from S-strain to R-strain bacteria pneumonial. Okay. One of the other scientists, which is Avery, and his colleagues, this scientist also, he made a lot of experiments. He made something awesome, something so good. What did he do? Actually, he makes something like isolating or separation. Okay, he isolated the virulent bacteria, okay, as a material that was already capable or suitable for inducing genetic transformation in the non-virulent bacteria. Okay, you get the what did he do? He isolate the virulent bacteria, okay, that already are induced by the genetic transformation to the non-virulent bacteria. The chemical and physical analysis showed something here, that this material is DNA. So the first sentence that we have, which is Griffith, he already gave us a final answer or evidence that the bacterial genetic or any DNA, it has ability for transformation. But he didn't give us the justification or explanation about what happened for this transformation, or what happened for the genetic material to be transformed. While Avery and his colleagues, they give us a final answer about what? About when he has ability to isolate this genetic material. So he found that the physiological or physical and chemical material, they already proved that this material is what is DNA. The bacterial transformation experiment can be explained as follows. Number one, a strain of bacteria observed an amount of DNA of another strain by a method, but this method till now isn't unknown. Okay, so the first step that we have, this strain or this type of bacteria, it already has the ability to observe amount of genetic material or DNA from another type by unknown method. Yet, they also has the ability to gain the characteristics of the bacterial strain from which DNA came. So, it has ability to observe amount of DNA and also to take the characteristics and proper features of the gained DNA. The characteristics of the DNA which this type of bacteria observed it. And also, more important, that this type of bacterial transformation or that received the DNA was transferred to the next generations. So, after observing or gaining these characteristics of the DNA, this type of bacteria has ability to transfer and to pass this information to the next generation, okay, to the next offspring. And here also we have crucial experiment, one also of the smartest experiment in the DNA genetic information field. An objection was carried out that DNA is a genetic material. Right now, we have how many experiments? We have a graph experiment, but he didn't give us an explanation of how the bacterial genetic information transformed, while Avery and his colleague they already proved for us that the transformed genetic information, it was DNA. And finally we have crucial, and he proved that the DNA is a genetic material. Okay, let's start with this experiment. Some which are based on that the DNA already it used was hot pure enough, but the contamination with protein, and this experiment, as I told you, it's awesome, or it's a very smart. Okay, perhaps this protein, which is exposed to or subjected to the temperature, was transforming material. Some scientists thought that really the transferred material, it wasn't DNA. Sometimes they thought, or they put this thinking as that this transformed, it was protein and it wasn't a DNA. But however, crucial experiment was carried out when an enzyme, this type of enzyme, has ability or capable of hydrolyze DNA completely, was isolated. Hydrolyze is mean what? To decompose DNA to its components, to its structural unit. Okay, so as soon as he bring and isolates an enzyme, and this enzyme has ability really to decompose or to analyze the DNA completely into its components, into its structural unit. So he concluded that this enzyme, which is called deoxyribonucleic hydrolysis of DNA, it completely, but it doesn't affect the proteins or RNA. So after isolating the material that or the unknown material, which has ability for transformation, this enzyme has ability to decompose it, and this enzyme is already convenient and appropriated for deoxyribonucleic acid. So they concluded that the DNA is already the genetic material that carry the genetic material information. It was found that when the transforming material was treated with this enzyme, or they subjected to this enzyme, it's already completely completely decomposed into its pure material, or its structural unit. The transformation didn't happen, and indicating that DNA is the genetic material. But we have another experiment to prove that the DNA is the genetic material. Bacteriophages. Well, bacteriophage is a type of viruses which has ability to infect also a specific or definite strain of bacteria. This virus particle consists of a molecule of DNA, okay, and a protein code. Well, actually, let me to give you here an extra information. We have two main types of viruses. The virus, actually, it might be just a part of DNA, single strand or double strand, or maybe RNA. And on the other cases, these viruses, sometimes they are protected or they are covered by a kind of coat. Okay, so we have two types. One of them is naked viruses, and the other type which is coated or enveloped. Okay, the enveloped viruses, they are consists of DNA, or just a part of DNA, okay, or maybe DNA double or single strand, and also it's covered or enveloped by a protein coat. This type of viruses, which is called bacteriophage, it consists of DNA covered or coated by a protein coat or protein envelope. Well, the bacteriophage used in this experiment was known to consist of DNA molecule and a protein coat, as we said, but which extend to something like a tail. So I'm going to show here a picture for it. We have here head, or the main part, which is the genetic material of the virus itself, and we have an extension which is something like, or it looks like a tail, okay, which attaches to the bacterial cell. It just an organelle, or its main function to fix the virus on the bacterial cell. It was observed that after 32 minutes of the attachment, or after the infection bacteria by this virus. So we have here a type of bacteria and virus. This virus called bacteriophage. It has ability to infect and to inject its DNA to the bacterial cell. Okay, so we have two parts of we have two parts here, or 2 points, bacteria and virus. The virus called bacteriophage. And what's going to be happen after the infection, or after the attachment of bacteriophage to the bacterial cell? After 30 minutes of the attachment, the bacteriophage, okay, it really has ability to inject all his genetic material to the bacterial cell. The bacterial cell burst is mean what? Explode. It absolutely disperses. It's already decomposed and analyzed, and comes out of it about more than 100 new complete another bacteriophages. At the end of the episode, I hope you get much more information about the DNA and the importance of studying the history of the DNA, and how some scientists prove that the DNA is the genetic material in the human and all living organisms. See you the next time.